Hey there, pre-AP. We're to our 4.3 outline in our molecular genetics unit, and let's talk about some DNA control mechanisms. So make sure you got a highlighter and a pen. If you lose your highlighter, then you just feel free to underline things with your pen or pencil. You'll be okay, I promise. Alright, so first thing, let's talk about operons. Now this is a new term. Operons. Please underline that is in prokaryote cells only. Out beside where it says prokaryote cells only, put bacteria. Just to remind yourself, we're not a prokaryote. We're not a bacteria. This is in bacteria only. Only. So let's highlight some things before we draw this. It'll make a little bit more sense after we draw it. So let's go. Operon system. Highlight that, please. This is an operator. Remember, operators help things. So we need to know that there are these two guys that discovered this. You don't have to know. It was the last name, Jacob and Madan. They discovered this in 1961. What we do need to highlight is an operon, that number 2. Operon, or operator, controls. Used to, back in the day, you had an operator that you could call, and they could switch you to call. Maybe you wanted to call the grocery store and see if they had a sale on ground beef. You would say, "Operator, could you connect me to the grocery store?" And they would say, "Sure." They controlled where your call went. Well, if you're a bacteria, you have operons, and they control how RNA polymerase has access to the DNA strand. Please out beside put that. Remember, you have an RNA polymerase and transcription. So just to keep you in mind what part of protein synthesis we're in. We're in transcription. And please highlight that an operon. What is an operon? Well, it's a part of the promoter sequence. Remember, promoter is that start here. Remember, he's the guy with the little flag, checkered. It's start here, start here. Don't start over there, start here. On that DNA strand. Start here. RNA polymerase, start reading right here. And we need to know that it is located between something called a TATA box and a start codon, just in case you get asked that sometime. We will not draw the TATA box or the start codon. It would confuse you. We're just going to make a vague drawing that will help you out. Alright, also know, number 4, you can have things that make transcription stop or start. If you stop transcription from happening, we say that you turn off the operon, like you turn off the lights with a light switch. Or you can turn on transcription. You can turn on the operon, like you turn on the light switch. So know number 4, if you are a repressor or a co-repressor, these are molecules that turn off or switch off transcription. They turn off the operon. Therefore, transcription can't happen. Or you're number 5. Let's highlight it. You're an inducer. You act as an on switch. You turn on transcription. So let's draw this, shall we? Alright, so let's draw an operon. You can put this at the bottom of your notes or on the back, whichever one you feel comfortable. So just put the word operon. We're going to draw an operon. So let's first make a very long segment. Remember, operons are going to be found on DNA, so if you need to put out beside there, this goes on DNA. The first part, we're going to divide this up. The first part is actually going to be a promoter. Let's actually spell that right. My mind's getting ahead of my pen. promoter. The second part is actually going to be your operon. This is where it's located. It's kind of behind the promoter, and over here somewhere would be a data box. Don't worry about what a data box means. You really don't have to know that. Now, after operon, you're going to divide this up into 3 sections. This is going to be gene 1, gene 2, gene 3. These are the segments that we actually want to turn into mRNA. We want to transcribe them. We want to transcribe them into mRNA. We want to take this DNA, these genes, because genes are just a section of DNA, and turn it into mRNA. So this is where an operon is. Now, let's talk about a repressor for a second. A repressor is going to be something. Let's change our pen color. We'll change it to, let's go with purple. A repressor, remember, this is going to turn off. This is an off switch. You've turned the lights off. It would be, you could have a shape like this. Let's put, and he's actually going to come and sit down on the operon. So let's name him repressor. He's going to actually come and sit down, so let's write that down. He sits on the operon. And if he sits down on the operon, what's going to happen? He turns off transcription. So that means that mRNA is not made. And then, in turn, something else is going to happen. If you turn off transcription, can you go on to translation? Oh, no, you can't. You can't. You got to go through transcription before you go through translation. So if you stop transcription, then you have stopped protein synthesis dead in its tracks. It can go no further if you're a bacteria. This means that no amino acid is made, and you can't make a protein. So remember, this repressor, it's an off switch. We are turning transcription off. It sits on the operon, turning transcription off. That means no mRNA is made, and that eventually means no amino acid is made. Oh my goodness, it's not good. But let's look at a different situation, shall we? Again, let's put the word. This is going to be your second example. So if you want to put operon number 2, this is not going to be a repressor situation. This is going to be an inducer. Let's see how you can turn on transcription. Let's see how that works. We're going to actually use a specific one in bacteria. It is called the lac operon. That's the name of it. It's an actual real operon. It happens in bacteria all the time. And we're going to draw it. So if you want to, let's draw it again. Let's draw our operon. We're going to make a really long piece. Again, we're going to put promoter. And then, after the promoter, that's right, we have an operon. Then, after operon, we're going to put that we want some genes in here. Maybe this time we have gene 1, gene 2, gene 3, gene 4. Now this is the lac operon. Lac being short for lactose. Lactose is a sugar. So usually, if you are in this lac operon situation, so this is actually the operon, usually in a bacteria, you already have a repressor sitting on you. It actually already sits here and actually has this shape. It actually is kind of a U shape. And it has a U shape for a specific reason. So it's usually sitting on top of the operon. And so you could put repressor. Usually there. It's usually here. Unless, this is the only way you can get that repressor to go away. You come and introduce some glucose molecules, or pardon me, lactose. So go ahead over here, put introduce lactose. You're going to introduce lactose, and this is what is going to happen. This little guy is going to be filled with a glucose, or pardon me, a lactose molecule. I'm used to saying glucose. And because it is been introduced to lactose, then your repressor is going to be removed. Now, why was it removed? Because lactose showed up. That's all it took. It just took some lactose to show up. It filled in that little horseshoe space, and then you can put repressor, removed, in presence of lactose, and transcription is turned on. Lactose over here is an inducer. Remember, inducers are turning on switches. Because lactose showed up, he was able to fill in the opening space of that repressor. And because it filled in that space, it could not fit on the operon anymore. Because it can't fit on the operon anymore, well, it's been removed. Now, RNA poly can now come and sit down on the promoter and start going through transcription and reading those genes to be transcribed. It couldn't do it before because that repressor was there. But because lactose was introduced, it came and filled up that space, removed that repressor because it didn't fit on the DNA strand anymore, and transcription is then turned on. Now, let's say if you don't have any lactose present. You don't have any lactose present. There's none to be found. None to be found at all right now. There's none present. Well, do you think that this is going to be able to go on and perform transcription? No. Only in the presence of glucose will this operon ever be induced or turned on. This repressor will always stay here. It'll stay there as long as it willy-nilly wants, because there's nothing to come and fill in this space. You fill in that space, repressor has to leave. No filling of the space, repressor stays. So only whenever lactose is introduced can you fill in the space and your repressor leaves and transcription is induced or turned on. So you might have a dataset question about a lac operon tomorrow. So keep that in mind. And I really hope that was helpful. Tomorrow, I'm just going to tell you what to highlight for Roman numeral 2, DNA control mechanisms in eukaryote cells. Transposons, we might watch a short video on tomorrow. They're kind of hard to understand unless you watch a little video. So, let's go with Roman numeral 3 right now, for DNA control mechanisms in eukaryote cells. So let's go with, you're going to find this in big letter A. Let's highlight. DNA is wound up. Like for mitosis, remember, wound up can also be called compressed, and it's not able to transcribe. Meaning that the transcription factory, that RNA polymerase, can't be built because it can't get access to the DNA strand. It's squished up. Your DNA is not in a nice pretty line like in the picture we just drew. But let's go ahead and highlight big letter B. If DNA is unwound, it kind of looks like spaghetti if you want to put spaghetti out beside there. Like in G1 of interphase, it is able to be transcribed. So, you can't transcribe anything while you're in mitosis because your nice little chromosomes are nice and compacted. This is what happens in mitosis. They're nice and compacted because you want to be able to move them around easily for cell division. But if you're like in interphase, like in G1, you can pick out a segment of DNA that you want to transcribe into mRNA, and it's okay because it's nice and loose. Also, for letter C, let's highlight that you can control this also by removing of introns and rearranging the exons in that translation modification. And then lastly, let's talk about the difference between chaperonins and rough endoplasmic reticulum. Why do we need one of those in a certain situation? So find that 1 and 2. Proteins, they get folded by that chaperonin. Remember that big old coffee cup I used? Chaperonins stay inside the cell. Those proteins need a chaperonin because they're going to stay inside the cell. They're going to be either an enzyme or they help with the cytoskeleton, the skeleton of the cell. Or, if you're a protein and you're going to go to the rough ER, you're going there because you're going to exit the cell. You're going to be used for either communication or in that ECM, the extracellular matrix. Now, why is this important? We need to highlight that Roman numeral 4. Genes that are transcribed, made from DNA into mRNA, help determine what the cell will mature into over time, what they're going to be when they grow up. So let's highlight where it says we call adult cells. We want them to become specialized or differentiated, which means all that means is highlight they carry out a special or different function, which kind of gives the question. If you're a cell, if you're a protein in a cell, what do you want to be when you grow up? Do you want to be an enzyme? Do you want to be a protein in the cell membrane? What do you want to be? Eventually you'll find out, depending if you go to the chaperonin and get folded up or the rough endoplasmic reticulum. So, we'll talk about jumping genes tomorrow, and I hope that was helpful, guys.